Two cucumber plants, same seed packet, same soil, planted on the same day in the same row in the same garden. 6 weeks later, one of them is barely 2 ft tall. The leaves are yellowing at the edges. The stem is thin. There are maybe 3 sad cucumbers hanging off it, and one of them is already rotting at the blossom end. The other plant, the one right next to it, is almost 6 ft tall. The leaves are dark, deep, jungle green. The stem is as thick as a man's thumb, and the vines are so heavy with fruit that the gardener had to drive 2 extra stakes into the ground and rope them up because the weight was pulling the whole trellis sideways. Same seed, same soil, same sun, same water. The only difference between those two plants is one liquid brewed in a kitchen for under $1 in the time it takes to watch this video. By the time we are done, you will know exactly how to make it, exactly how to upgrade it into a complete fertilizer, and exactly why the chemical industry cannot put this on a shelf, even if they wanted to. So, stay with me, because this is the version most people on the internet are not showing you. Most gardening advice gets the order wrong. People obsess over leaves, over fruit, over color, over what they can see above the dirt. The plant does not work that way. The plant builds from the bottom up, roots first, microbial relationships first, soil chemistry first. Everything you see above the ground is a downstream effect of what is happening underneath. If the biology underneath the soil is weak, no amount of expensive fertilizer poured on top will fix it. You are just feeding a broken engine more fuel. What we are about to make solves that at the root, literally. It does not feed the plant directly. It feeds the living organisms in the soil that feed the plant, and then it doubles down by releasing growth hormones, instant amino acids, and stress protection compounds directly into the root zone. The result is what you saw in those 2 cucumber plants. Not a small difference, a categorical difference. The kind of difference that makes neighbors stop and ask what you are doing. The hero of this recipe is a single-celled fungus called Saccharomyces cerevisiae. If that name sounds intimidating, relax. You already have it in your kitchen. It is the scientific name for ordinary baker's yeast, the same yeast you use for pizza dough, the same yeast you use for bread. Most people think of yeast as a one-trick organism. Add sugar, it makes bubbles, your dough rises. That is the entire story for most people. That is also a tiny fraction of what yeast actually does. Yeast is a fungus, and like every living fungus, the moment you give it water and sugar, it wakes up and starts producing a chemical cocktail that is wildly more complex than a few bubbles. Peer-reviewed research published in Crop Science, Agronomy, BMC Plant Biology, and Frontiers in Plant Science has documented 4 separate ways that yeast directly transforms plant health. Let me walk you through all 4. This is the part that turns the recipe from a folk remedy into something you can defend with science. The first mechanism is hormones. Saccharomyces cerevisiae produces 2 of the most important plant growth regulators in the natural world: auxins and cytokinins. Auxin, specifically indole-3 acetic acid, is the master signal that tells a plant cell to stretch out and divide. It is the molecule responsible for new root growth. Every time a seedling pushes a new root tip into the soil, auxin is the chemical telling it where to go and how fast to grow. Cytokinins do the same job above the soil. They tell cells at the growing tips to multiply rapidly. They are the chemical reason a plant can put on 6 in of new growth in a single week. The rooting powder you buy at the garden center for $15 to $20 is just a synthetic version of auxin. That is the entire product, a laboratory chemical that mimics what yeast produces for free. When you pour fermented yeast water into your soil, you are flooding the root zone with both of these hormones at the same time. You are not asking the plant to grow. You are telling it to grow with the exact chemical instructions it would normally have to produce for itself. The second mechanism is what I call the instant meal. Plants normally absorb nutrients in two ways. They can pull mineral ions directly out of the soil water, which is slow, or they can wait for microbes to break down organic matter into amino acids and peptides, which is even slower. Yeast skips both lines. As the colony multiplies, it releases amino acids, peptides, B vitamins, and trace minerals directly into the surrounding water. These molecules are already broken down. The plant does not have to digest anything. The plant does not have to wait for anything. The roots simply drink them in. It is the difference between handing someone a raw steak and asking them to grow muscle from it and running an intravenous protein drip straight into the bloodstream. One takes days, the other takes minutes. This is also why you see the explosive growth response in 72 hours. The plant is not slowly converting nutrients. It is absorbing pre-built nutrition the moment it touches the root zone. The third mechanism is the immune shield. A study published in 2014 in the journal Crop Science documented something fascinating. When yeast cells are introduced into the soil around plant roots, they do not just feed the plant. They colonize the root zone. They occupy the physical real estate around the roots. This matters because soil is a battlefield. Pathogenic fungi such as Fusarium and Pythium and root rot bacteria are constantly attacking weak plants. They enter the root zone and begin consuming the plant from below long before the gardener notices anything is wrong. Yeast outcompetes those pathogens. It crowds them out, and it occupies the space they would have invaded. It also releases compounds called glycopeptides that trigger the plant's own internal immune system to switch on, the same way a vaccine teaches your body to defend itself. Plants treated with yeast solutions show measurably lower rates of fungal disease, root rot, and damping off in seedlings. Your plant does not just grow faster, it grows defended. The fourth mechanism is the one most gardening channels skip entirely, and it might be the most important: drought armor. Saccharomyces cerevisiae produces a compound called trehalose. Trehalose is a non-reducing sugar that yeast manufactures to protect its own cells from drying out. It is the same compound that allows desert organisms to survive years of desiccation. When yeast colonizes the soil, trehalose ends up in the root zone. Multiple peer-reviewed studies, including a 2023 review in BMC Plant Biology and a major paper in the Journal of Plant Growth Regulation, have documented what happens next. The plant absorbs the trehalose. Once inside the plant tissue, it acts as an osmoprotectant. It regulates how the plant opens and closes the tiny pores in its leaves, called stomata. It controls water use efficiency at the cellular level. The practical result is this: when a heat wave hits, when your neighbor's plants are wilting, drooping, sunburnt, dropping fruit, yours stay standing. Yours keep their color. Yours keep producing. The trehalose in their tissue is regulating water loss far more efficiently than untreated plants ever could. This is not theoretical. Researchers have transferred yeast trehalose-producing genes directly into tobacco, rice, and maize, and watched the plants survive drought conditions that killed the controls. You are getting a softer, simpler version of the same effect just by watering with fermented yeast. So, here is the obvious question. The same question you should be asking every time something this powerful exists for under $1. If yeast water is this effective, if it does 4 different things at once, if it is backed by half a dozen peer-reviewed studies, why is it not sold in a bottle at every garden center on Earth? The answer has 2 parts. Part 1. It physically cannot be bottled. A chemical fertilizer is dead. It is mineral salt dissolved in water with a stabilizer. It sits on a shelf in Arizona for 5 years at 110 degrees and nothing happens to it. A living yeast culture is the exact opposite. The moment yeast touches sugar and water, it starts eating. It produces carbon dioxide. It produces alcohol. Pressure builds inside the container. If a manufacturer tried to bottle this and ship it nationwide, the bottles would either explode in the warehouse or arrive at your house already spoiled. The few commercial live culture biostimulants that do exist sell for $40 a pouch, require refrigeration, and expire within months. Part 2. It kills the entire business model. You only need to learn this recipe one time. After that, you are out of the market forever. You don't buy rooting hormone. You don't buy seedling starter. You don't buy probiotic soil drench. You buy yeast at the grocery store next to the eggs for under $1. There is no patent. There is no profit margin. There is no subscription. There is no way for a corporation to insert themselves between you and the result. So, we make it ourselves. Listen carefully now, because what I am about to give you is the longer fermentation master version. If you already know the quick 30-minute brew, this is the upgrade. More yeast development, more biology, more potency. 1 quart of warm water, not hot, not cold, around body temperature. If you stick a finger in and it feels neutral, you are right. If it feels hot, you will kill the yeast before it ever activates. If it feels cold, the yeast cells will stay dormant. 1 2 of dry active yeast. And here is a tip. Expired yeast often still works. Half-expired packets from the back of your pantry can still contain live spores. Do not throw them out. 2 TBSP of granulated sugar. White, brown, or raw. It does not matter. Remember, the sugar is not for the plant. Plants have no use for raw sugar. The sugar is the fuel that wakes up the yeast colony and tells it to multiply. Combine all 3 ingredients in a glass jar. Stir until everything is fully dissolved. Now cover the jar, but do not seal it tight. The fermentation will produce gas. A sealed lid can pop or shatter. Use a loose lid, a paper towel secured with a rubber band, or a folded cloth held with string. The goal is to let the gas escape while keeping insects out and blocking ultraviolet light from damaging the active yeast. Place the jar somewhere dark and warm. A kitchen cabinet, a pantry, a garage that does not drop below 60 degrees. Now wait. For the master version, you are waiting between 2 to 12 hours, not minutes, hours. The longer you wait within that window, the more the yeast colony multiplies. The more biology you build. The more hormones, amino acids, and trehalose accumulate in the liquid. You will see a thick, foamy beige cap form on the surface, exactly like the head on a freshly poured beer. That foam is the visible proof that the colony is alive and active. When you see that head, your concentrate is ready. Now the most important step, dilution. Do not pour this directly on your plants. It is far too concentrated. You will burn fine root hairs and shock the soil microbiome. Take your 1/4 fermented concentrate and pour it into a standard 5-gallon bucket of plain water. Rainwater is best. Dechlorinated tap water is fine. That gives you a 1.20 dilution, the safe ratio for direct soil application. Stir well. The bucket is now your finished living biostimulant. Now, here is where this video goes further than every other yeast water tutorial on the internet. The base recipe handles biology and root growth beautifully, but it is light on certain critical minerals. Potassium for fruit development, magnesium for chlorophyll, and calcium for stem and cell wall strength. You can fix all 3 with items already in your kitchen and your fireplace. This turns the recipe from a growth booster into a complete biological superfeed. Additive number 1. Wood ash. If you have a fireplace, a wood stove, or a fire pit, you probably have one of the best potassium sources on Earth sitting in a bucket somewhere. Wood ash is roughly 5 to 7% potassium by weight, plus calcium, magnesium, and trace elements. Add 1 TBSP of clean wood ash to your 5-gallon bucket. Do not use charcoal, painted wood, or treated lumber. Potassium is the mineral responsible for big, heavy, sugar-rich fruit. This is what turns small tomatoes into giant tomatoes and small peppers into thick-walled monsters. Additive number 2. Epsom salt. Add 1 TBSP of Epsom salt, magnesium sulfate, to the bucket. Magnesium sits at the center of every chlorophyll molecule. Without magnesium, leaves go pale yellow even when the plant has plenty of nitrogen. With it, the leaves turn that deep, almost waxy dark green that signals a plant operating at full photosynthetic capacity. Additive number 3. Eggshell powder. Take your dried eggshells, grind them as fine as you possibly can. A coffee grinder works perfectly. Add 1 TBSP of the powder to the bucket. Eggshells are nearly pure calcium carbonate, and the fine grind gives the soil microbes the surface area they need to break it down quickly. Calcium builds strong stems, thick cell walls, and most importantly, it prevents blossom end rot in tomatoes, peppers, and squash. If you have ever lost a tomato to that ugly black bottom, this additive alone is worth the entire recipe. Stir all 3 into the bucket with the diluted yeast water. The yeast biology will actually help unlock and chelate these minerals, making them more available to plant roots than they would be from any commercial fertilizer. Now you have a 5-gallon bucket of living biology, growth hormones, drought protection, immune support, potassium, magnesium, calcium, and a full spectrum of amino acids and B vitamins. All of it brewed from kitchen scraps, all of it for under $1. Pour it at the base of your plants. Soak the soil thoroughly. Do not splash the leaves with this version. The minerals can leave residue. Apply it at 3 moments per season. First, at transplant to trigger a root explosion. Second, at the start of flowering to hold flowers and prevent drop. Third, once when fruit first appears to meet the heavy nutrient demand of fruit fill. Within 72 hours, the leaves will stand straighter and color up. Within a week, you will see visible thickening of the main stem. By week 3, you will start to see the categorical difference, the kind that produced the second cucumber plant in our opening. The yeast biology in this recipe pairs with one specific soil amendment that almost no gardener uses correctly. When you stack them together, the results stop looking like normal gardening and start looking like commercial greenhouse production. I broke down the full method in another video, and it is sitting on your screen right now. Click it next, because this story is not finished until you see that one, because the gap between an average garden and an unbelievable garden is not money. It is not luck. It is not even hard work. It is knowing the recipes the industry buried. Brew the potion. Fortify it. Water your plants tonight. And then watch the next one. The harvest begins now. If this story changed the way you look at the simple ingredients in your pantry, please subscribe and like the video. We are uncovering the natural secrets that the big corporations have tried to keep hidden. Let's grow a better future together. The next garden treasure will be revealed soon.